So I'm not a clinician like Tom, I'm a physiologist. I do the experiments and I try to find the evidence. I'm not a theoretician. For me, evidence is experiment. So I have a different take on this whole area, and I want to approach on precisely what it is that we're talking about. Because Glycocalyx has got this sex appeal because of its name, Glycocalyx, like vitamin, interferon. So everyone is now having the tendency to project all their problems onto the Glycocalyx, which is probably why most of you are here. So the issue that I'd like to talk about today is there is the idea that the Glycocalyx is an important component of the vascular barrier. And is that true? And is there evidence for that? How do the slides look?
Yeah, the green one is your pointer.
So, as mentioned, I'm a physiologist. I head a department in the Academic Medical Center in Amsterdam. I'm also a physiologist in the Department of Intensive Care, where I work 2 days in the week in Erasmus Medical Center. And so my job is to translate physiological concepts to the bedside. So, or is it? Is the Glycocalyx maybe nothing to do with vascular barrier? So, first of all, it's important to, I think, look at the component of the Glycocalyx in its integrity. So, from a physiological point of view, it is important to take the whole organ, as it were. So, we are not proponents of reductionist attitude to looking at what physiology is doing. Just taking out the Glycocalyx and studying it and expecting it to give you answers without taking its surroundings, without taking the blood components, without taking the endothelial cells, smooth muscle cells, etc. And from that point of view, if you take the microcirculation as an organ, you really see, and sepsis is the most extreme example, really, of a hit for the microcirculation, you see that a whole bunch of stuff is going on together. And in a lot of these experimental models, when they apply these enzymatic treatments to these organs, they are destroying a lot more than just the Glycocalyx. So I think it's important to realize that any type of intervention in physiology translates itself into many other components not functioning, and to then directly assume that this is a single component responsible for a single pathology is anti-physiological, as I hope to show to you. So one of the most important things is really the disassociation between the systemic and the microcirculation in disease states. And another important issue is that therapy used clinically is really completely, totally focused on manipulating systemic circulation. And the expectation is that this will translate itself to an improvement in the tissues. Completely no evidence for that. So blood pressure is being manipulated for what? Well, if you ultimately take the clinician through his way of thinking, you always come down into tissue perfusion and transport of oxygen to the cells. That's the bottom line. That's why you give fluids, presumably. That's why you give blood. That's why you give vasopressors, etc. So that is a very important understanding. And the Glycocalyx, in that particular context, its function is important to understand. So if we look at the Glycocalyx from a functional point of view, in that what is it exactly doing to the physiology, one of the most important components, and Dr. Woodcock already mentioned that, is some work by Weinbaum and co-workers showing that inside the Glycocalyx there are these mechanotransductors which are able to transform shear stress of the red blood cells into nitric oxide production of the endothelial cells, and in that way regulate dilation or constriction of the blood vessels depending on the needs, metabolic needs. So this is of course a classic picture from a physiology book showing the filtration and the oncotic pressure and reabsorption, which we now learn does not exist. Nevertheless, it's what we teach our medical students. Also, the barrier function of that vessel being an important component of this particular image. So the clinical problem that we're talking about is not really glycocalyc degradation, it's this. it is capillary leak syndrome. That is really the reason why people, clinicians, are attracted to the concept of the glycocalyx, because they think that understanding the glycocalyx will tell them something about capillary leak and tissue edema. And this was really one of the first descriptions of capillary leak syndrome, now called the Clarkson syndrome, in a premenstrual lady started to become extreme edema. She finally died and went into shock. Repeatedly, she started to get tissue edema, and the question really was what this was. And this is the patient that we're talking to, and that's why glycocalyx is of such interest to clinicians, because they feel that it will tell us something about this patient and the fluids, etc., and the compromised capillary bed. So if we now look at the vascular barrier in general, yes, the glycocalyx is an important component. So of course is the integrity of the endothelial cells themselves. There are fenestrations, there are pores, there is all sorts of leaks in the different endothelial cells. And what's important, which hasn't really been talked about too much, is that it's extremely heterogeneous in the different organ systems, and also within the organ systems. So the degradation and the buildup of the glycocalyx is also very heterogeneous. In the liver, it's being degraded a lot more quickly than, for example, in the kidney or the lung. So an important component really also, not talked very much about, are the junctions in between the endothelial cells. These are the adherents, the cadherents, but also important signaling molecules. So we now know that endothelial cells are connected to each other by gap junctions, that membrane potentials are being transmitted upstream through the endothelial cells, and is actually an important component of the control of upstream vasotone. So physiologists have always been surprised that if you downstream drop acetylcholine, then you can get upstream vasodilation. And it has to do really with membrane potentials going through the endothelium upstream. So one of the most important actually aspects is turning out to be of vascular regulation. So it's a very complicated system, and when talking about the glycocalyx, in my opinion, you have to take the whole system into account. You can't just look at one of the components and then make inference about the whole system, and that's what we're going to do. So one of the things that we have introduced, and some of you may know, into the field over the years are these handheld microscopes with which we can observe the microcirculations of patients at the bedside. It was the first time we introduced it in '99, and we're able to image the brain microcirculation. This was during surgery, and in the meantime we've applied it to cancer. This was a study looking at nitroglycerin in septic shock patients, and we've looked at it in various other applications. You can see here these images in which you see dark with white. We basically use the optical contrast of hemoglobin as an agent which is absorbing green light, and that way we can look at functional capillaries in which there are red blood cells. So everything you see as dark is in fact red blood cells. We can't see non-filled capillaries. So this is the latest device that we have used, and we can actually see single red blood cells going to capillaries in great detail. Especially in intensive care medicine, this technique has made an enormous impact because people all of a sudden saw what adverse effects all the things they were doing routinely really have at this level. Some 600 papers have been published. There's some 300 to 600 of these devices worldwide at the moment. So this should work. Can you maybe press the button or something? Yes. So this is what a septic patient looks like. Now if you see this image, you immediately understand why there's an oxygen extraction deficit in sepsis. Because here we see vessels that are perfusing next to vessels that are completely obstructed. We understand why you can have high venous saturations in the presence of high lactates. We understand a lot about the pathophysiology of shock actually when you see these type of images. And we also understand that if you start giving fluids to this patient, it actually becomes worse. Because the fast bits start becoming faster and the clogged up bits just remain clogged up. And it makes us understand that really just giving fluids is nonsense if this is the type of issue that you're trying to treat.
But could you say that again, please?
It's going to get
fluids is nonsense, because I mean that's the bottom line of what you're talking about. That's why this is so important.
The complete sentence was, Paul, giving fluids on its own is nonsense if you want to correct this type of
Exactly.
pathophysiology. Yes.
Exactly.
Absolutely, yes.
Exactly.
So, you need a soup, basically. A soup.
I mean, because
It's a different lecture, but
what you're saying
chicken
is
soup
so
maybe.
fundamentally important, because if you read the approach, the first approach to resuscitation and sepsis is give fluids.
Of course, but you know that better than I do, that the surviving sepsis guidelines
Yeah, but I
should really
mean,
be
the
ignored.
problem is people think that that's the truth.
I know, but I'm just a physiologist, so
Yeah,
I'm just
but I
telling you
mean,
what
so
the
that's
evidence
what you're
shows.
saying, actually, from a physiological, but it's just
Okay.
so important.
So let's continue on that happy note, if I can say so. We got together in Colombia, people interested in kidney, actually, the ATKI group, and we wrote a paper on endothelium and sepsis. And of course, the glycocalyx is a very, very important point from a functional point of view. So we see the glycocalyx really as a cabinet, considering some important molecules for normal physiology, for homeostasis. So the mechanotransduction, the presence of antioxidants, Van Miliband's factor, and also a very important issue to realize is the adhesion molecules in the glycocalyx. So the glycocalyx is a relatively thick structure, and the adhesion molecules are very thin, very small. So it's when the glycocalyx actually becomes is shedded that these adhesion molecules come to surface, and that's really why the leukocytes then can stick and roll across the across the endothelial surface. So from our perspective, the glycocalyx really is a very, very clever structure which contains a bunch of molecules as an adaptive response to inflammation. And that's it. So from that perspective, it contributes to the barrier, but it's not key to the barrier, was our basic idea. So if we now go quickly through the glycocalyx, this has been discussed before, there are these images. What is also important from a hemodynamic point of view is to realize that if you start to shed the glycocalyx, which is this distance in between here, you're actually going to rise in capillary hematocrits. So that too is an important factor. So the concept of anemia from a microcirculatory point of view is very uncertain when you start messing about with intravascular volume. This is a very nice paper from Weinbaum in Reviews of Biomedical Engineering showing the mechanotransduction function of the glycocalyx, and many experiments have been done too. This is a particular nice paper showing that if you have an intact glycocalyx and you control the perfusion, that nitric oxide production is released, which is a one-to-one reason how the smooth muscle cells are influenced to actually regulate vasotone. And if you take it away, you see there is no relationship. If you take the glycocalyx away, there's no relationship between shear stress and production of nitric oxide. So it's a very important signaling element when it is intact, and it is the basis and foundation of autoregulation. So if you actually take away the glycocalyx, as occurs in critical illness, autoregulation is one of the first to go. And if you give fluids, you destroy the glycocalyx, and you also destroy autoregulation, which we know. So again, as Bernard very elegantly showed in his studies, there are a number of techniques, all pretty complicated lab techniques. I will just skip through them because these have already been handled. What is an important issue is to realize that these the glycocalyx are structured in certain areas. One of the interesting parts, indeed, is that in the kidney they are piled up, as it were, in the fenestras. So we always thought that the fenestras were just a sort of like a window. It's the word, Italian word for window. But actually there is a thick glycocalyx in that particular fenestrations. And the fenestrations are directly related to the filtration coefficient of the different organ systems, in which, of course, they're the least fenestrated organ is the brain, for example, and the most fenestrated are the of the endothelial cells are in the kidney and in the lung. So we also know that reactive oxygen species are really a very, very effective way of destroying the glycocalyx. There was a question about that before. You do anything in oxidative stress and you destroy it. So, and reperfusion injury is probably the easiest way to induce a burst, as it were, of oxygen radicals, which destroys the glycocalyx. And you can protect your patients against this by loading them up, not after the damage has been done, which is the clinical way to do it, but prophylactically before, for giving antioxidants like vitamin C, but in this particular case, superoxide dismutase. So if you manage to reduce the oxidative stress load prophylactically, you can protect the glycocalyx. So this was another one of the techniques that Bernard talked about. We had used it in 2005, which is really looking at the escape rate of tracers of different size molecules. We looked at it in a model. We were looking at hyperglycemia, and again a very interesting result from this, in which acute hyperglycemia caused oxidative stress and a degradation of the vascular barrier, because you're not really measuring the glycocalyx here, you're measuring the vascular barrier. But then if you had chronic hyperglycemia in a diabetic model of a mouse, you actually, the compensatory mechanisms were able to deal with that hyperglycemia, and that it is really the acute hyperglycemia which is a dangerous one, which is a different story. But anyway, this was a study that we did. So in one of my questions, I think it was to you, Bernard, this is a particularly imposing paper. It's like, you know, whenever papers appear in the New England Journal of Medicine or Nature Medicine or papers like this, you have to wait until it gets reproduced in the real journals, like the American Journal of Physiology or the Journal of Physiology, before you start believing it. And this is one of those, oh wow, things, looking at the important role of heparinase in the pathophysiology of ARDS. And they're actually suggesting that a heparinase knockout mice, and then you get the survival. If you have a wild type, it dies, and if you don't have the heparinase, you survive. And heparinase, that was the real hit for the glycocalyx in causing the ARDS. And it is certainly true that sepsis causes an upregulation of heparinase, and that heparinase could be involved, but this translation exactly, this paper has never been reproduced. And I know people who've tried to reproduce this study and have been unsuccessful. So as Tom already mentioned, now people who have never even seen a glycocalyx, have never done any research on the glycocalyx, don't even really know what it is, start reproducing these type of sexy pictures, sort of explaining all, and people say, oh my God, look at that, that's terrible, with arrows going in, and here bushy ones with arrows going the other way, and all of a sudden in New England Journal of Medicine, so it must be true. This has really polluted the literature, in my opinion, on the meaning of the glycocalyx. So now we see these type of studies which try to connect two different areas. And this is one such study, albuminuria, which we know is a clinical issue, problem. And again, what they do here is they give endotoxin, which basically destroys everything. And they see that the syndican 1 levels go up, and they see that the albumin goes up, and they say, okay, the glycocalyx degradation is causing the albumin to go up. But actually it is, you've destroyed the kidney, which is why the albumin goes up. And certainly the glycocalyx is also, but this specific role for the glycocalyx in determining the vascular barrier for syndican is certainly not demonstrated by studies like this. This is a very nice study, Bernard. I don't know if you've seen this one. It's just appeared in Critical Care, which is a Japanese study. I think they use, now this is not my area at all, but nitrate staining. And here you can see very nicely a healthy kidney, the inside of the glomeruli, without the staining, and with the staining, you see these sort of broccoli-like structures, which is the glycocalyx. And this is what happens in the septic kidney. You see this complete destruction, really, not only of the glycocalyx, but really of practically everything else in the kidney. So when you look at studies relating glycocalyx and sepsis, be very careful about interpreting the studies, because sepsis just destroys practically everything that you can imagine. So too was this connection with edema and the protection of the glycocalyx. And this was a study done by Bernard himself, standing here, first author. And here you can see before and after the treatment. Again, the specificity of this compound for just destroying the glycocalyx still has to be demonstrated, and probably is destroying a lot more than just the glycocalyx on its own. What is very nice in this particular study is a demonstration of the edema by measuring the distance between the capillaries. So a larger distance showing more edema. And you'll also see this rise in capillary hematocrit as a result of the degradation. So you start to have a self-perpetuating idea that glycocalyx is directly related to vascular permeability. So we decided to investigate this specific issue about the relationship between glycocalyx degradation and vascular barrier function. So this is Philippe Guerci from Nancy, who spent a year with us, and this is Bülent Ergin, our postdoc, and together they did a study looking at the vascular barrier permeability glycocalyx in hemorrhagic shock and heme dilution. We also did reperfusion injury and everything that you can imagine. So basically, we used every technique that we were able to apply to look at vascular barrier and endothelial glycocalyx function. One of the ones was the ejection of the different fluorescent dyes, in which we then repeatedly looked at the extravasation of the different size molecules. We also did a series of experiments using Evans blue, the so-called Smurf rats, in which we give Evans blue and then look at the extravasation of Evans blue into the tissues. It's a standard physiological model for looking at vascular permeability. And this was basically our shock model. So I'm just presenting you the hemorrhagic shock part of the study. Our idea was do different types of fluids protect the actual glycocalyx and, in its extension, the vascular barrier. So this was basically it. We gave the animal a hemorrhagic shock. What's very important in this study is to realize, and this is really the reason why, in my opinion, the whole area of fluids is such a mess in critical care and anesthesiology, is because clinicians do not appreciate the importance of resuscitation endpoints. So people give a liter of this and a liter of that, but they never target a hemodynamic endpoint. And this is the reason why people do not really know how to use colloids. I'm a great fan of colloids, let me be clear about that. Because if you actually target a hemodynamic endpoint, you use a lot less. And the less you use, the better it is. So anything, in my opinion, which allows a clinician to use less fluids to correct some hemodynamic abnormality is good. So if you have a real hypovolemia, in my opinion, you need colloids, because with less colloids, you can correct that stage for a longer period of time. However, if you have dehydration, and unfortunately a lot of clinicians do not know the difference between the two, then using of colloids is completely meaningless. But using crystalloids, especially if it's intracellular dehydration, without glucose is equally meaningless. So there is this mess in fluid land because people do not take hemodynamics into consideration when applying fluids. It's a different thing. People talk about the composition, they don't talk about the amount and the timing, etc. But that's my own thing. In any case, we do in this study. So basically we bleed the animal, then we resuscitate the animal with different types of fluids, and we target a mean arterial pressure of 80 millimeters of mercury. So that's a targeted shock. And at the end here, we give these different boluses of, we inject the fluorescent dyes, and then we sample them at different time points and look at the decay curves. So this is real shock. I mean, here, look at the lactate values, the 5, 5, 5. I mean, there's no question about that. These animals are in shock. Reperfusion injury equally. Lactates go up. And if you give these animal fluids, you indeed are able to correct the lactate, so everything that you would expect to happen with giving replenishing volume does indeed occur as you would with all the different types of solutions. So now you see the advantage of using the different composition. If you use your endpoint, you see that you need a lot more saline to reach your resuscitation endpoint of 80 millimeters of mercury than if you use a starch molecules. This is the reason why you need colloids in South Africa to treat hypovolemic traumatic shock patients. It's as simple as that. And this is time and time again. You see it. It's about 2 to 3. What's also interesting that if you look at balanced crystalloids, in comparison to saline. You also need less balanced crystalloids to reach the same hemodynamic targets in comparison to saline, about 2 to 1. And we've shown this also in many, many studies. So, again, saline, it's really not a very good solution, although a lot of people don't think it is. So, what happened with tissue edema? Wet weight, dry weight is the golden standard. No effect. These animals, even though they were in complete shock and received a lot of fluids, no tissue edema in the liver, in the kidney, in the lung brain, irrespective of the type of fluids that we use. So what about the retention, the vascular barrier? No difference between any one of them. Here you can see the different decay curves. There was nothing wrong with the vascular barrier of these animals, even though they had been in deep shock. And this was true for the immediate septic shock, for the heme dilution, and for this hemorrhagic shock. What about the Evans blue? No difference between the different ones. These are completely different techniques. So we took the animals and we did intravital microscopy, which is, people are asking, what do you think is the gold standard? This I think is the gold standard, at least for vascular barrier. Here you can see actually the vessel, and so we look at the seepage of the dye outside the vessel. This is the only time we managed to get the dye outside of the vessel, and that's when we turned the heating pad on to 42 degrees and cooked the animal, and basically then we were able to get some leakage of the dye. Again, no difference using direct fluorescent microscopy. What about the glycocalyx shedding products? All of them went up. And there was really no difference between them, with the exception maybe of the hyaluronase in which the balanced Hess solution was statistically lower than the crystalloid solution. But otherwise, all of the fluids in the resuscitated phase were shedding the glycocalyx, but there was no change in the vascular barrier. So what about the microcirculation? Now, if we look at it from a theoretical point of view, if you say, okay, what's the microcirculation showing us? Well, if you disrupt the glycocalyx, you do have an effect, theoretically, on microcirculation, because the lumen is going to change, it's going to become stickier because of the leukocytes, etc. And indeed, we looked at muscle microcirculation, here you can see, and here you can see, indeed, if you look at the flow, this is the mean flow, you see a reduction, especially in the saline group after the resuscitation. So even though the vascular barrier was not affected, the microcirculatory flow kinetics, the hemodynamics was, and the glycocalyx was too. So from a physiological compartment point of view, this all makes completely sense. If you take away an important component of the endothelial cells, you disrupt the hemodynamics. It does not translate itself into a vascular barrier, and so despite microcirculatory derangement, glycocalyx degradation occurs. We see that there's no significant alterations in the vascular barrier. And are the results similar in different models? The answer is yes, because we did the experiments. So how must we then look at these glycocalyx? Well, what's interesting is that some of the compounds in these glycocalyx have got really good things which are happening. For example, this interesting study looking at Sindocan-1 found that it really was very effective in the clearance of pro-inflammatory chemokines. So the components of the glycocalyx also have benefit in terms of controlling inflammation. Not only the leukocyte trafficking, but also the components have got interesting positive effects, promoting in our idea that the glycocalyx degradation is an adaptive response to inflammation in this. So what other components could be associated with vascular leakage? Because that's how we started, capillary leak syndrome. Well, let's look at these adherents and cadherents, and Lee and Slutsky wrote this paper in New England Journal of Medicine. Interestingly enough, I asked Arthur Slutsky about this. I said, well, I didn't know you guys did any work on adherents and cadherents. He says, I've never seen it. We've never done it. New England Journal of Medicine asked us to do the review. We did the review, but we've never looked at it. So this really shows, again, unmasks the comedy of the New England Journal of Medicine, in which they ask people who've done nothing about it to write reviews on a subject like this. Nevertheless, very important, a real expert is, of course, Neil Granger from Louisiana, who's a guru in reperfusion injury land, and he has written this very nice paper, really, about the importance of these adherents and cadherents in a host of different diseases. And if you look at what they actually do, so these are these adherents and cadherents specifically, and they're anchored to actin-myosin filaments inside endothelial cells. Very interesting structures, beautiful immunohistochemistry studies about that. And if you actually give them, which is cytomix, which is also sort of a septic cocktail on them, you can actually see the disruption of these intercellular locations. And if you actually look at humans too, you see, especially in lung specimens, specifically in the capillaries, specifically in the lungs, that these adherents and cadherents become destroyed. And you can actually measure them also in plasma, which is a very interesting, I think, biomarker to look at, because they get cleaved, and you can look at soluble antibodies against these adherents and cadherents. Here we see a study which has been done in sepsis, in which you see them being upregulated. And I don't know why I've got this. Oh yes, and just going on back. And here again too, you see that the upregulation of these adherents and cadherents and the dysfunction of the glycocalyx is associated with the leukocyte rolling, which the paper that Bernard had showed earlier had also demonstrated. So there is a function. So we said leukocytes, that's great. So using our microscope, here you can see very nicely, this is a cardiac surgery patient. Here you see the white chaps, the leukocytes. As you follow them carefully, you can see them here. They stick and they remain in the tissues. So this is a technique. Here you see them. I don't know if you can see those two balls that are sitting there. So here you've got a technique which allows you to make the connection between hemodynamics and inflammation at the bedside in a patient. And can you protect the glycocalyx? Yes. Hydrocortisone, vitamin C, they're the ones. So these are ones I dedicate to Paul Merrick in the back. Beautiful study by the group in Munich, looking at the protective effects of steroids and also vitamin C treatment, reducing capillary leakage prophylactively again. And we have looked at vitamin C in great detail in a number of studies. Here we've got leukocyte infiltration being, this is a ischemia reperfusion model, being reduced by vitamin C. Tissue MDA, which is a marker for lipid peroxidation, also being reduced very significantly with this. So in conclusion, the glycocalyx is not a key component of the vascular barrier. Its protection of vascular function is achieved by anti-inflammatory and antioxidant drugs such as vitamin T and limitation of fluids. So that's my presentation to you today.